Yeah. Now the first speaker of the morning, this morning, is Erwin Schoof. I hope that I'm saying the name correctly. Okay, here it is. I see him. Okay. Can you hear us?
Yes,
Yeah.
can you hear me?
Oh, okay, good. Yeah, we can hear you.
Excellent.
I mean, the day is going to be pretty full, so for the sake of time, I'm going to say very, very few words about each of the presenters for the sake of time and also because I'm not good at all at presenting people. I took a few notes. Erwin is from associate professor at the Technical University of Denmark. Yesterday, I went through your website and I noticed that you are working in a number of interesting projects. I've seen single cell signaling in leukemia, for example, and that's something that, is, triggering my curiosity. So we are really looking forward to your presentation. So please go ahead, share your screen, and thank you very much again.
Great, thank you. I assume you can see my slides at the moment?
Yeah.
Yeah, perfect.
Yeah, go ahead.
Well, thanks for that very kind introduction, and of course for the invite to having me present here today. It's a real pleasure. I wish I could have been there personally, but I have a lot of travel at the moment, so apologies for that. But indeed, I'm fortunate to earlier this year have started my lab called the Cell Diversity Lab here at DTU, and indeed we're really keen on investigating various complex biological systems with single cell perspective, both at protein and genome level. And today I really want to talk about how we're applying single cell proteomics and transcriptomics in the context of human hematopoiesis. So I basically mainly want to focus on two things today, kind of give you a brief background on the human blood system and how we are doing single cell proteomics, and to then give you the story so far of how we've been applying single cell multiomics on human bone marrow. So just to get everybody on the same page, as I'm sure many of you in the audience are familiar with the human hematopoietic system, it's really been a system intensively studied through various means. And I mean, it's a hierarchical system. So we have our long-term hematopoietic stem cells up here at the apex that differentiate into short-term hematopoietic stem cells. And then we have the myeloid and the lymphoid lineages. And that's kind of been the de facto standard of how we look at this system for probably even decades at this point, and very nicely summarized in these papers here. And kind of the more we study this system, the more we realize that it's actually much more complex than we originally thought it was, that this, upper stem cell population actually consists of many more populations. But really, since we now have access to single cell technologies and especially transcriptomics at this point, we actually now have been able to change this dogma and moving away from kind of this notion of having discrete cell states to it rather being a developmental continuum, and that catching these cells in very discrete cell states is more or less impossible, and helps explain why we had trouble finding real specific markers for some of these populations, because they are continuously differentiating or dividing. And to be able to study these trajectories, we really do require single cell resolution technologies to be able to figure out how these cells decide to go from a stem cell to, for example, an erythrocyte or a B cell or a T cell, etc. So as said, a lot of this is based on transcriptomics. There's been huge efforts done in the field, and I think they're most nicely summarized in this Azimuth reference map that you can find online. It's basically a public database, and in the case of human bone marrow, there's about 300,000 single cells here where they've done single cell transcriptomics and are indeed able to pinpoint these key populations that are contained within the human blood hierarchy and that we can use to refer back to in order to help explain our data. But I'll get in much more detail later on in the talk. So of course, since we are today talking about proteomics, I also wanted to kind of touch on this notion of how are we going to move from an mRNA perspective to a protein perspective, and why should we actually? And I'm probably preaching to the choir here since this is a mass spec meeting, but I mean, we all know that, of course, mRNA can act as a proxy for protein expression, but there's various functions that are affecting this relation, like protein translation, protein degradation, and of course, with RNA-seq, there's no way that we can actually capture any of these protein modifications, such as phosphorylation or acetylation. And this is really where the protein level study becomes important. This is something that I've had a big interest in kind of throughout my academic adventures. So if you want to read up on some of these applications, here are some manuscripts. But of course, to understand protein level single cell functioning, we need the technology for this. And this is really why my group basically got interested in how do we determine cell heterogeneity at the protein level, to which end we set out to develop global single cell proteomics approaches that we call single cell mass spec. So, I mean, I think indeed it's safe to say that the persons that started this whole notion of, yes, we are able to do single cell proteomics at this point were Bogdan Budnik and Nikolai Slavov, and Nikolai will be presenting later today with their approach called SCOPE-MS. And this is a really clever technology because it uses tandem mass tag technology, which is basically a mass tag where you can add on unique reporter ions on this end to be able to distinguish which sample the signal came from, to be able to both multiplex single cells, but importantly also to be able to add a carrier or a booster channel to facilitate peptide identification. So if you, for example, take a 200 cell equivalent sample, you label that with a unique TMT tag, especially the 126, you would see in your mass spec spectrum, in an MS2 spectrum, you would see a signal belonging to this carrier channel, which of course will be much more intense than your actual single cell signals. But by labeling the proteins and peptides coming from your single cells with TMT, we can distinguish very clearly the carrier channel and the individual single cells. And from these reporter ions, do single cell protein quantification. Whereas, of course, the more higher MZ value peaks are the peptide fragment ions that help identification. And this is really the technology that we were very excited about, both in terms of throughput and proteome coverage. And you may have seen our paper earlier this year, where we basically tried to optimize on this method by including the so-called real-time search algorithm that's implemented on the Orbitrap Eclipse. So what real-time search does is that, you know, when we get our MS1 scan, the instrument will pick a few peaks, do an MS2 on them in the linear ion trap, which is very sensitive, and therefore we need only very little injection time, 23 or 46 milliseconds. And it'll do on the fly a real-time search of that MS2 spectrum. And only those spectra that it could identify into a peptide would it then trigger this long injection time MS2 or MS3 scan in order to quantify and identify these single cell peptides with very high accuracy. Because of this carrier channel, we need to inject at very long injection times. And through the reticle method, we're not wasting any scans that take the 750 milliseconds if we were never going to identify them, like in the case of these first two peptides. And that's really, I would say, the revolution of the Eclipse and now the Ascend, that you have this real-time search to allow you to spend time on those peptides that matter. So we investigated kind of MS2 versus MS3 quant, and then also, of course, the standard MS2 without any real-time search at a variety of injection times, so for half a second or 750 milliseconds. And indeed, what was nice to see was that through the real-time search, the MS3 performance in terms of numbers of proteins identified per cell is basically as good as MS2 standard level analysis. But what we were very happy to see is that our reticle method actually allowed us to boost coverage to about 1,000 proteins per cell at a throughput of around 10 samples per day. And due to the TMT multiplexing, we can now run 160 cells per day per instrument using this technology. If you want to know more details about this, it's all contained here in this manuscript. Of course, single cell gives a lot of data, so we also had to figure out how to process all this data and to kind of automate this as much as possible. And here, Benjamin, a PhD student or joint PhD student in my lab, he built this pipeline called Scepter, which is very closely integrated with a leading tool from the RNA-seq field called ScanPy. So we basically put our raw files through Proteome Discoverer. We use fact sorting to deposit our single cells, where we also collect the surface markers through index sorting. And then we collect all the results in Python. We process the facts data using FlowKit, and then our Scepter pipeline will do cell annotation, cell normalization, filtering for quality control, as kind of shown here, that those where we had too much signal because probably 2 cells were deposited, we remove. We can remove empty wells, and we can basically set cutoffs based on either signal to noise here on the x-axis or number of proteins here on the y-axis to really make sure that we only contain wells that pass a certain quality control. As a general problem in single cell, we have a lot of missing values. So Scepter will also allow you to do k-nearest neighbors imputation to allow adding more protein identifications across many cells to make better clustering of your cells. And this is kind of shown here. So the normalization is very important because each injection has a batch effect on its own due to the TMT pro-multiplexing. So if we don't do any normalization, you would basically cluster all your cells simply by injection on the mass spec. But through this normalization function implemented in Scepter, we're actually separating out our cells according to cell phenotype rather than sample run on the instrument. So we remove most of the technical batch effects through this normalization approach. So then to switch gears and now start talking how we're applying single cell proteomics and transcriptomics. I really want to stress that this is really the work of many people. We have 4 PhD students. So Benjamin and Neil are joined between my group and Bo Porce's group. Despina, who just got her PhD a few weeks back, used to be Bo Porce's PhD student for the RNA-seq. And we're working together with Sabrina from the Fabian Theis lab to help us integrate this data together with the single cell transcriptomics. So as mentioned, we have this huge azimuth reference data set, and I just wanted to point out that we're not sampling the CD34 negative population because these are basically fully differentiated cells, and we're really trying to study the more primitive part of the hierarchy, i.e. the upper part with the stem cells, and then down to these myeloid and lymphoid progenitors. And for those of you familiar with FACS data and FACS sorting, basically this is your first gate where we separate cells according to CD34 and CD38. And then if we take this bottom gate, the 34 positive, 38 negative, here we have our HSCs, our multipotent progenitors, and the long-term multipotent progenitors, or the so this is really the stem cell compartment, whereas the double positive 34 and 38 positive populations can then be segregated by 2 additional gates with CD45RA and CD10, where we have our CLPs. And if we take this bottom gate and we further segregate those cells based on 45RA and CD123, we can separate the CMP, the MEP, and the GMP cells to add basically enriched cell populations to our analysis. And this entire kind of part of the hierarchy is commonly referred to as the hematopoietic stem and progenitor cell, or HSPC compartment, that we decided to study here. So Despina basically took care of doing the site-seq analysis, where we, from 4 individuals, so basically healthy people that came and donated some of their bone marrow, we subjected about 10,000 of these HSPCs to site-seq transcriptomics, where we found about 4,000 highly variable genes, whereas NIL took care of the single-cell proteomics, where we subjected about 3,000 HSPCs from 6 different people. We recorded 10 FACS markers during the collection of these cells. We identified about 3,000 proteins in total, with a mean of 950 proteins per cell. So kind of a first step to see if we're capturing any of this heterogeneity between these cell types, we wanted to just draw a HuMap. And you can see that in the single cell sequencing, we get very nice kind of clustering of cell types, and they're annotated here. But actually, even in the proteomics data, we are capable of separating out our cell populations according to the proteomes measured, because these UMAPs are drawn based on these 3,000 proteins that we measured, and they're clustering according to cell type rather than by any arbitrary batch effect, which we were very happy to see as a first kind of quality control. But of course, we wanted to dig much deeper into this. So first of all, as mentioned, we integrate our FACS markers into our analysis, so we can basically relate back the intracellular proteomes that we measure on the mass spec back to the FACS markers that were measured during the collection of these cells. And this is just to showcase how we annotated these populations then in Scepter. So here again, we have our stem cell, our HSCs and NPPs on the 34-38 gate. And then we draw quite stringent gates around them, so that, for example, these cells here that are in between, that we don't mix up our cell labeling by these transitional states, and rather have more pure populations that we annotate. And you can see that here as well, that these MEPs are really a stringent gate drawn around them to not confuse these cells in the middle that we basically just leave unannotated and ask later on what the proteomes are telling us these cells are. And this is really just to apply stringent gating to reduce any ambiguity we might find from these cell labels. So a first sanity check then is, of course, to validate that our clustering based on proteomes measured in the mass spec correlate back to the FACS markers that we know these cells should be expressing. So for example, CD45RA over here, we know that should be GMP and CLP. So if we look at which cells light up with the CD45RA, we can indeed see that it is the CLP in red and the GMP in pink that are expressing CD45RA. If we look at, for example, CD10 and CLPs are the only cell population in our data that express CD10, we can see that again, we have this very nice correlation here in red with the CLPs lighting up as CD10 positive in the FACS markers. Another nice control was this endomucin, which is an HSC marker. You can see that it's mainly the HSCs down here, also the MPPs down here that are expressing this endomucin, suggesting again that the clustering based on the proteomes measured can be correlated back accurately to the FACS markers. So this was a really nice first quality control and sanity check that our data makes any sense at all. And that indeed we are able to recapitulate this HSPC hierarchy. What about this donor to donor variation? Because as I mentioned, we collected bone marrow from 6 individuals and subjected them to our workflow. And you would expect that each individual would kind of spread across this HuMap quite evenly. And we were very pleased to see that this was indeed the case. You can see that some individuals, we ended up with more cells than others. But very critically, we see that all individuals are spread across these entire UMAPs, suggesting that it's actually reproducible from what we're seeing. And for those of you with a keen eye, I'm listing here young and old, and that's basically young people versus slightly older people, which we were hoping would reveal some key key differences between young and old bone marrow, but this far we haven't gotten yet with the data analysis, but we hope to report on that in the near future. But just to conclude that the donor-to-donor variation appears minimal, allowing us to integrate this data set into one big data set of about 3,000 single cells. In terms of the site-seq, this was really where Sabrina used her knowledge and experience in doing site-seq data quality control. So here she annotated the cells with azimuth, integrated all the individuals with total VI, and you can read up on these methods in these papers. And we could see that the data actually follows the correct trajectories with the HSCs at the apex and then going out into the various branches that we know exist. Also here, the 4 individuals that we sampled, and these are the same individuals as the single-cell proteomics, also spread nicely across these HuMaps, suggesting again that the transcriptomics data, as expected, is also recapitulating the hierarchy nicely, and that all the individuals are following similar trends in terms of their cell populations. So then we were wondering, well, can we use the fact that the mRNA data is so rich, and can we learn something from the mRNA and then port that back onto our proteomics data? Simply because there's so much more knowledge out there from transcriptomics than proteomics, we figured we should see if we can learn something from those resources. So if we here have our single-cell RNA-seq data annotated with azimuth, we can extract certain markers for these individual populations, i.e. RNA transcripts that correlate with, for example, an HSC or a Ba-IOMA population, and so forth. But then we overlaid those lists with proteins that we also found in our single-cell proteomics data. And that kind of led to this short list of let's say, 10 markers that are cell type specific according to these populations. So if we then ask, well, how do these RNA markers that we also found in our single-cell proteomics data, how do they cluster or visualize or separate the cell types in RNA space? And we can see, for example, MPO down here, which would correspond to the GMP, indeed very nicely marks this GMP population down here. The histone 1 ends up lighting up here, and this is actually where our stem cells are. So it does suggest that indeed histone 1 FO is a stem cell specific gene marker. CLC down here, this would be a Ba-IOMA population as listed here in blue, suggesting that indeed these markers truly separate cell types at the transcript level. But then, of course, the big question is, do they also separate cell types in protein space? And this is what we then set out to validate, and we're actually very pleased to see that indeed histone 1 lights up down here, which in protein space are our HSCs and MPPs. So indeed confirming that histone 1 is an HSC marker in our protein level data. Here we have MPO, which is a GMP marker, and indeed our pink GMPs here is exactly where we see this protein show up. CLC, which was the Ba-IOMA marker, that's very highly expressed in this cluster here, that we at this point we're still calling CMP, but I'll get to how we can re-annotate these populations using these markers. So it does really suggest that from mRNA markers, we can also re-annotate protein level populations. So by using these mRNA markers for these cell populations, we can recluster our data. So this is the same data, it's just now re-annotated with these new markers extracted from azimuth, where we can now call these cells Ba-IOMA instead of CLP, or CMP, sorry. So the CLPs here that used to be in red, we now know that this was actually 2 populations. So the CLPs are subdivided into a CLP population, but also in pro-B and pre-B here in gray, meaning that we now suddenly get more resolution of these cell populations than we had with the proteomics data alone. So indeed, GMP are now subdivided into GMP and pre-dendritic cells. And unfortunately, we were not able to reconstruct cell markers for CLP or MPP, and basically attribute this to those cell types really being transition states and therefore don't have any key proteins that distinguish them. So far, we've been able to establish that RNA level data helps us re-annotate our proteomics level data, but can we actually integrate these data sets and make it one big data set full of cells determined by both RNA and protein? So this is a very recent analysis. This is all very much ongoing work, and we hope to wrap it up relatively soon. So I just wanted to share some preliminary results with you. So here we use a glue machine learning environment to basically generate this RNA and protein correspondence that can guide the integration to do this joint embedding on a UMAP of protein cells and RNA cells, i.e. cells that were measured with proteomics or with transcriptomics. And lo and behold, Sabrina and Benjamin were able to do this and basically build one massive UMAP of all the cells. And if we now highlight all the RNA level cells, you can see still this clear distinction between cell types and colors. But even if we highlight our protein level cells, we still get nice clustering according to these populations, meaning that it suggests that the integration actually works. If a cell is, for example, a bi-IMaSS cell in RNA space, also our protein level data would annotate it as a bi-IMaSS here in red and here in blue in the RNA space. So if we overlay this to kind of make it more clear how well these cells are actually integrating, we can see indeed that they cover the full UMAP and suggesting that all our single cell proteomic cell types overlap well with the azimuth annotations, and that we can now really do this label transfer from RNA cells to protein cells. And why is this useful, you might ask? Well, it allows us to start looking at cell trajectories, because in the end, we're interested in what decides whether an HSC turns into a GMP or a pre-B cell or a bi-IMaSS cell. And for that, we need to look at trajectories. And the Fabian Theis lab just published a cell rank paper earlier this year, which is a very good computational tool to infer these trajectories. So we wanted to test, does this work well on this joint embedding of RNA and protein? And lo and behold, it actually does. So if we have our HSCs down here, we can see this trajectory going up to GMP here. And then our GMPs are highlighted here. And the late erythroid branch ends up being annotated here. So we're really seeing these nice trajectories showing up on these integrated embeddings. So this was a good first sanity check that computationally we can now find these trajectories, and now we just need to dig in to the data much more, find these known gene expression changes, and see if we can find some correlations between site-seq and FACS markers. So the last thing we can now do is ask, well, how much do RNA and protein actually agree? So on this plot, I'm just showing examples for two proteins, EEF1A1 and MPO, and then separated either at RNA level or at protein level in the bottom row, and then across trajectories from HSC to bi-IMaSS to GMP to late erythroid, pre-B1, and so forth. And you can see that, for example, MPO agrees very nicely between RNA and protein. But for example, this EEF1A1 is very high in the RNA in an HSC and then stops being expressed as you differentiate into a bi-IMaSS. Whereas at protein level, we can see that it's the opposite trend. So this is really how we argue that single cell proteomics will help us improve the readouts from RNA-seq by truly figuring out what's being expressed in these cells. So we now need to do this much more globally, but I just wanted to share these two examples to kind of show, that some agree, some disagree, and that we now have the tools in place to explore this in much more depth. So just to conclude, I hope I convince you that single-cell proteomics, while still in its very early days, is actually able to start tackling biological questions, both in terms of cell heterogeneity, but also cell differentiation pathways. That multimodal analysis of single cells is beneficial for accurate cell type clustering. And I really think, given the current scaling capabilities, i.e. transcriptomics can measure up to, let's say, 8,000 genes per cell, and we can run a 10,000 cell experiment very easily. Whereas with proteomics, we're limited to about 1,000 proteins per cell and 100, maybe 200 cells per day, so much lower throughput. So if we integrate this single-cell RNA-seq data appropriately, we can boost the data quality of single-cell proteomics. This integration is challenging, but also appears to be equally rewarding. So now we're looking at this differential expression analysis across these different clusters to find clear cell markers. And then hopefully in the future, we're going to be adding diseased samples such as acute myeloid leukemia or precursor states like MDS. Of course, we're very interested in being able to do sequencing and proteomics from the actual same single cells, and we're working on technology to do that. And then, of course, also to do this in a spatial context, in solid tumors, etc. So with that, I'd really like to thank especially Benjamin and Nil, who've really pushed the bone marrow work, Bo Porse and John, who have been fantastic postdoc mentors before I started my own group, the Fabian Theis lab, of course, various companies that we work very closely with, the DGU Proteomics Core, my other group members, our funding agencies, and of course you for your attention. Be happy to take any questions if there's time.
Thank you very much. Super interesting. I think we have time for a couple of questions for Erwin. Sure. I hope so. Wait a second.
Hello. Okay.
Yeah, I think you're.
Thank you, Erwin, for this super nice presentation. I just wanted to ask, what do you think about your carrier channel and how should it be composed? Should it represent like the single cells as closely as possible? And how do you design your carrier?
Great question. It's something we looked at a lot in the early days because indeed we thought, do we need to mix kind of equal ratios of the different cell types? I mean, the short answer is that it doesn't seem to matter too much. As in, we now ended up taking just CD34 positive bone marrow cells for this entire experiment. Because we argued that would be the most kind of universal carrier. And it was able to find these subpopulations. It seems to confirm that biology is not very binary. It's not like proteins are off or on, but it's rather more kind of they're up or down regulated to some extent, meaning that in theory, we should be able to pick them up in the carrier channel as long as it's a heterogeneous mix of those cell types. I mean, in our leukemia work that we published last year, we were actually doing this one-to-one cell mixture of the 3 cell types we were looking at in that paper. It did help to find a few cell-specific markers, but so it probably depends on the question you're trying to answer. Like if you're trying to find cell populations, it's actually fine to use a kind of heterogeneous booster channel. But if you really are keen on finding very specific markers expressed only in a very low percentage of cells, I would probably recommend to make a more clever design of your booster or carrier channel to be able to pick up those proteins so they don't drown in the other high abundant proteins contained therein.
Thank you.
Hi, Erwin. Here's Carl. Great talk.
Thank you.
Enjoyed. I have 2 questions. The first one is the beginning of the presentation. You pointed out in average, you found 3,000 proteins per cell, 950. But if you have in one analysis 950 cells, you have something it's called missing channels. How do you overcome the problem in multiple runs with TMT, the missing channel problem? Because if you have 10 or 100 cells, you feel down to 10% of all proteins what you found in all runs together.
Yes. No, you're absolutely right, Carl. It's a huge problem. Luckily, it's not unique to proteomics. I mean, 10x genomics also has this probably around 40% missing values. So we use this k nearest neighbor imputation as implemented in Scepter. But of course, we're not imputing, 2,000 proteins in every cell, because if we're measuring 950 proteins in every cell, but measure 3,000 across all the cells, if we add 2,000 fake measurements, imputed measurements, we're going to be distorting the biology quite a lot. So we rather put in certain filters, and this is very data set dependent, but we set requirements to have measured a given protein in, let's say, 10, 20, 50 cells before we then allow that protein to be imputed in the rest of the cells. So this is where we end up with probably about 2,000 proteins in total represented in a UMAP, out of which, half is imputed and half are real measurements. But this is where the TMT approach is indeed not optimal. I think we're going to see that with the data independent analysis, this might get much better. That's at least a hope. But yeah, so we can deal with it to some extent, but I agree, we should also get much better at decreasing this experimentally.
I absolutely agree with you, because you can impute data, but if you work with TMT, you never have idea what's the expression of the in the TMT if you don't have the fragments, yes?
Yes.
Mm-hmm. And the second question I have, I think it's quite interesting question. I was not sure that for your approach, you really need single cell proteomics, yes? Because you sort the cells, yes?
Mm-hmm.
But it was not clear for me what's the advantage of single cell proteomics? Did you find something unique in some cells or not? Or is this more for all cells?
Yeah, so if I understand your question correctly, you're asking what if we sorted these subpopulations in bulk, as in we sorted some GMPs in bulk, some HSCs in bulk, and then ran them as bulk to then compare the cell populations that way. Is that along the lines of what you're? Yeah. So we've done that. I mean, that was most of my postdoc where I did that, and we were having real trouble getting cell-specific markers coming out of that analysis. And I think this is also what we're now seeing from the single-cell transcriptomics, that these cells keep actively differentiating. So capturing a cell that we, according to the FACS markers, call a GMP or an HSC, these cells are probably already on the way to being another cell type, meaning that this heterogeneity within these FACS-based markers is quite substantial, and that we would not capture the true proteomic identity of these cells if we did this in a bulk fashion. And this is really where we need to do this at single cell level to be able to really capture this cell heterogeneity within each population and to be able to build the trajectories. We wouldn't be able to follow a cell from an HSC out to a GMP or a bio-IMaSS cell unless we can capture enough cells that are along that trajectory or differentiation trajectory to be able to figure out which proteins are expressed to cause this differentiation process to occur. Of course, the limit is the number of proteins we're getting. I mean, are we truly getting these cell regulatory proteins like transcription factors, epigenetic regulators? Not as much as we would like, but I think we will, and see with better sample prep, better instrument handling, both of which I'm sure you're going to talk about later, that we get this better depth to start getting to these low abundant key cell regulator proteins. That's when we're really going to see some magic.
Thank you.
Thank you.
Okay. Thank you very much again.